Now, Assalamu alaikum. Now we're going to start with the embryology lecture from where on we left. That is, we were talking about the menstrual cycle. That is, its two components. We talked about the two components, that is the ovarian cycle and the uterine cycle. So generally, I'll recollect what we exactly did. That at the end of this phase, we said when there are two conditions: when there is fertilization, what all is going to happen, and what is the end when there is no fertilization. When it is no fertilization, the process is as a whole that it starts from the hypothalamus, it stimulates the pituitary gland. Pituitary gland produce, and the interior portion of the pituitary gland, it secretes the gonadotrophins, that is the follicular stimulating hormone, that is FSH, and that acts on the ovary, and luteinizing hormone that basically acts when the second half of the ovarian cycle. This FSH is going to mature the follicle. You can see the different stages that the follicle from starting from the primordial follicle is reached to the mature Graafian follicle, and then there is that is roughly in the middle, that is the 14th day of the menstrual cycle, and there again luteinizing hormone started to act. This then produced the ovulation, and then after ovulation the ovum was released, and the rest, what is all left, was formed the corpus luteum. This produces the progesterone, which is going to act on the uterine endometrium. The thing is that the first half is producing estrogen, which is going to act on the endometrium of the uterus. That is, after menstrual cycle, after the menstrual phase, the uterine endometrium is just the basal layer that is left, that has to be rebuilt again. That estrogen is going to act as the proliferative phase. It's going to build up the blood vessels, it's going to build up the connective tissue and the endothelium. So at the middle of the level, you see that the level of the blood vessels, at the height of the blood vessels, the endothelium, that is nearly to what was there in the normal side. Then when the corpus luteum is produced, again it is the luteinizing hormone, or the secretory phase starts. That is, there is tortuous coiled blood vessels are produced, edematous stroma is there, and the epithelium that is already prepared. If it is a fertilized ovum, then it will embed in this edematous and secretory phase endometrium. Then the glands are dilated and tortuous, and so they're going to provide the nutrition to the expected entering is fertilized zygote. This is when there is no fertilization has occurred. Then if fertilization occurs, similar changes are there, but then this phase, that is the corpus luteum, that continues. Previously we said that corpus luteum is going to degenerate if there is no fertilization. So as you can see that corpus luteum finishes, and then again there is a withdrawal, and then there is menstrual cycle starts again. Then if there is fertilization, it is going to continue. The corpus luteum continues to produce the progesterone and maintain the endometrium, so that it is going to help the implanted embryo that occurs, that to grow until and unless the placenta is formed, which provides the progesterone, and then corpus luteum becomes corpus luteum albicans, and then that maintains the developing zygote. So that is the phase when the fertilization occurs. These are the two changes which are going to be there. Now we're going to talk about how these gametes, which are going to fertilize, they reach the respective site. We've talked that these gametes are produced, they are going to travel to the respective sites. So it is going to be the transportation of the gametes. It is oocyte transportation and sperm transportation. How do they reach? How does the oocyte reach? That is how the ovulation occurs. In secondary oocyte is expelled from the oocyte. We have already talked from the ovary. The stigma is produced, stretch of the ovarian wall, that relates to avascular area of that there, then that ruptures and it releases the ovum. At that time, the fimbriated end of the fallopian tube, that is going to cover the ovary at that site, and it's going to suck in by its current the ovary, the released ovum, and then that is going to be taken into the fallopian tube, that is the infundibulum of the uterine tube, and this passes to the level of the ampulla of the uterine tube, and that is further, it goes on by the peristaltic contractions of the uterus and plus peristaltic contractions of the fallopian tube. Then this is how what occurs. This is the picture, that is when ovulation is about to occur. The Graafian follicle is produced, it's going to rupture, or infundibulum covers it, and it sucks in the ovulated ovum. Then how does the sperm discharge? The sperm, when it is produced in the testes, that is stays in in the epididymis, a part of the testes, and then it is from transported to the urethra by the contraction of this part, that is its ductus deferens. If you see this portion, that is the testes. Testes are there covered by this tail type thing, that is the epididymis. They are going to be stored over there, and when it is going to be ejaculated, they are going to pass this duct. As you can see, it is labeled, it is the ductus deferens. It's going to pass through the ductus deferens, going around the urinary bladder, and it reaches or enters into the urethra. And this ductus deferens meeting the seminal vesicle gland, that is they become the ejaculatory duct. If you can see these two, the ductus deferens plus the duct of the seminal gland, they become the ejaculatory duct, and they open into the prostatic part of the urethra. Now urethra has three components. One part is, the first part is called the pre-prostatic part, that is before the part which enters into the prostate. Then it is the part which passes through the prostate, and then it is this membrane in between, that is the membranous portion, and then it is the penile portion of the urethra. So these gland, the sperm, they're going to enter into first into the prostatic part of the urethra. So by the contraction of these ductus deferens, they are going to reach into the prostatic part of the urethra. This is not purely the gametes which are there, it is the thing, something what we call as the seminal fluid, and that is going to form, and it's going to float in these seminal fluids. This is produced by the accessory sex glands. One is what we call as the seminal gland, that was, I saw you the picture, in the portion area next to the prostate, and the other is the prostate and certain bulbourethral glands which are around it. So in the area where you have the prostate, there are different glands. You have the seminal vesicles, you have the prostate, and you have the bulbourethral glands. All these produce secretions which are added up to the sperms, and they produce what is called the semen or the seminal fluid at this level. So the sperms which are coming from there, secretions from these three glands, they are added up and we form what is called as the semen. Then the semen has something what is called as prostaglandins, and it is said that at the time of intercourse, the uterine motility is increased, and that is done by these prostaglandins which are present in the semen. And the fructose which is there in the semen, that is an energy source for the sperms which are present in the seminal fluid. So these are the, so there it is entered into the urethral part of the prostatic part of the urethra. If you see in this picture, from the testes into the epididymis, they are stored. When there is going to be ejaculated, it passes into the ductus deferens. This duct which is there, then it goes round this ductus deferens. This enters into this part where you have the prostatic part of the urethra, where it meets the seminal gland duct, and they together they form this ejaculatory duct, and this ejaculatory duct opens into the urethra. Urethra, as I told you, the different portions: pre-prostatic, prostatic, the membranous, and then the penile part of the urethra. So it enters the seminal duct, the ductus deferens opens into this prostatic part of the urethra along with this, and so its semen is there. And then when there is going to be emission or ejaculation, then this is going to pass out through this penile portion of the urethra. Then there are two conditions. How does it ejaculate? Ejaculation of the semen is reflex ejaculation of the semen in two phases. First, it is emitted, that is, semen is delivered to the prostatic part from the testes to the prostatic part of the urethra, that is a sympathetic response. And then it is ejaculation, in that semen is expelled from the external urethral orifice. So ejaculation of semen takes place in two phases. When there is sympathetic response, it comes on to this prostatic part of the urethra from the testes along with its seminal fluid, and then ejaculation is through the external urethral orifice, that is the opening of the penile portion of the urethra. The volume of this semen is about 2 to 6 ml, and sperm speed varies pH of the environment. Non-motile in the epididymis become motile when there is ejaculation. They move slowly when they are in the acidic medium of the vagina, and when they reach the uterus, that is more alkaline, they become more motile. But it is also said that if the acidity is more in the female genital tract, then more chances of infertility are there, because these sperms, they die out and they cannot move up till the fallopian tube. 200 sperms reach the fertilization site. Most degenerate and are reabsorbed by the female genital tract. Only a few are there which are going to pass through the different processes, which is going to fertilize the ovum in the ampullary part of the fallopian tube. Then the sperm transport is about 200 to 600 million sperms are deposited around the external walls of the uterus during sexual intercourse. They pass through the cervical canal by the movements of their tail. Passage of the sperms through the uterus and uterine tubes is by the muscular contractions. There is an enzyme. Once they are lodged in the cervix, there in the vagina, there is an enzyme which is called the vesiculase. It forms a plug in the vagina and it prevents the retrograde flow of these sperms outside. When ovulation occurs, the cervical mucus becomes thin. That is, so they're lodged in the vagina, they're deposited in the vagina. There, this enzyme vesiculase is going to coagulate some of the semen so that it and forms a plug, and it does not allow the sperms to pass out. When ovulation occurs, the cervical mucus becomes thin, and it allows the sperms to move upward to the and reach the fallopian tube. Now, the process is that at coitus, that is at this process of intercourse, it is they are deposited in the interior vagina. They quickly contact the cervical mucus and they enter into the cervix, which we said is more alkaline in nature. Vagina is more acidic, and so it is chances that the motility would be less. So in the cervical mucus, it filters out sperms with poor morphology, that is, which are not normal, and motility. And they enter the cervix. In the uterus contraction, it passes the sperms passes upward into the fallopian tube. A few thousand they swim through the uterotubal junction, that is the junction of the uterus and the fallopian tube, to reach the fallopian tube, the contraction, and by the swimming they pass upward, and where they're at that junction, they are going to be stored as a reservoir. At the time of ovulation, as the ovulation reaches, these sperms have to penetrate. In order to penetrate the ovum, they have to pass through a procedure what we call as capacitated or capacitation and hyperactivated. Otherwise, they are unable to fertilize the ovum. So the sperm become capacitated and hyperactivated, which enables them to proceed towards the tubal ampulla, where the fertilization is going to occur. So motility, hyperactivity, as the sperm in penetrating the mucus in the tubes, and the cumulus oophrus and zona pellucida and oocytes are finally fused with that. So once the ovulation starts, the sperm has to change. It has to become capacitated and hyperactivity activated, which enables them to proceed towards the tubal ampulla, where the ovum is going to be there, and there they're going to meet together, and then they're going to pass through the procedure of this fertilization, which we'll talk about later on. So this is the process. Deposited in the vagina, then they are going to pass upwards into the alkaline media of the cervix, where they reside, and these sperms, they are filtered out with poor morphology and lack of motility, that is, which are not physiologically or morphologically, which are not normal. They are stayed behind. A minority of them are actually, they enter upwards, and they're going to go, and where you have the tubo-uterine junction, they are stored there. Once ovulation occurs, they are going to change, that is, they're going to modify, that is, capacitation is going to occur, and then they proceed towards the ampulla, where the fertilization is going to occur. It said only 1% of the sperm deposited in the vagina enter the cervix, where they may survive for many hours. They move to the uterine tube, a muscular contraction and propulsion of the sperm, that is their motility. Reaching the isthmus, sperm become less motile and cease their migration. The ones at ovulation, again, they become motile, and by these cumulus cells, and they swim to the ampulla, where fertilization is going to occur. So they reach the isthmus, that is the point of the fallopian tube, which is just at where it is the entry of the uterus. They are going to stay there, and the motility becomes less. Once ovulation occurs, again, the motility is going to increase, and then these sperm are going to be capacitated, and they enter in the cumulus cells. That is all the function of the fertilization starts in the ampulla. How long can these gametes, so this is the process from where the oocytes and the spermocytes, the sperms which are produced, how they are transported into the sperms, they are transported into the female tract, and how from passing through the male tract, and in the oocytes, how they are ovulated, and then they pass into the fallopian tube in the ampulla, where they are going to reside for fertilization. So these are the areas where the fertilization is going to occur, and this is the way the gametes are transported. Once they are produced male gametes from the testes and female gametes from the ovum. Then they have to be released to reach these areas, that is, they have to reach the fallopian tube. So, and then the male gametes, they're going to be entered into the semen, and by the semen, by the emission and ejaculation, they're going to be released, deposited in the vagina. Then they go up into the cervix, and from the cervix, they're going to travel to the isthmus of the fallopian tube. And once ovulation occurs, the motility again increases, and then they're going to reach to the ampulla. So that is the transport of the male and the female gametes. How long can these gametes survive? The human oocytes has said that they can usually fertilize within 12 hours after ovulation. They cannot be after 24 hours, they degenerate. And the human sperms, more than 48 hours in the female genital tract. So it's roughly 24 hours and double in the male sperms. Some sperms, at times they're stored or they are present in the folds of the cervix, and they're gradually released and to the cervical canal, pass into the uterine tube. So at times these can also produce the function of fertilization, increases the chances of fertilization. That means even if they have some methods used for preventing pregnancy, but these sperms which are already present, they can again be released from the folds of the cervix and reach the fallopian tube and do the fertilization. So sperms and oocytes can also be stored in the frozen procedures used for, and they help in the assisted reproduction can be there. Fertilization. So we've formed the gametes. They are both in haploid condition. We have transported them from the area where they were produced and from where they are going to reach the fallopian tube, and then at ovulation, when they are required, when there is the intercourse is there or the sexual relationship is there, then this is going to act together. The sperm and this reach the ampulla and fertilization is going to occur. Now, what is fertilization? Fertilization is the process by which a sperm cell and an egg cell unite to form a completely new individual. There are different stages of fertilization, that is, can be divided into 4 processes. One is sperm preparation, that is what we said as it was capacitation. It has to occur, that is, before fertilization, this process must occur. Then the sperm and the egg, they recognize and they come close to each other. Then they fuse together, and then the sperm and the egg fuse, and then they become large in size, and that is what we call as the male pronucleus and the female pronucleus, and this is the activation of the formation of the zygote. So these are the different steps which are going to occur or which are going to take place when the fertilization has to occur. There are 2 types of fertilization. One is external fertilization, in which the sperm cell unites with an egg cell open rather than inside the specialized organ, and the other is the internal fertilization inside the female after insemination or through copulation. So that is normally what occurs in humans. Then the thing which I said is capacitation. Spermatozoa are not able to fertilize the oocyte immediately upon arrival in the female genital tract. As I've said, they reach the isthmus of the fallopian tube, but they are not able to penetrate the ovum or the layers which are outside the released ovum, but they must undergo 2 reactions. What is capacitation, and the other is the acrosome reaction must occur before they meet the female gametes, that is the oocyte. These 2 must occur. These are the prerequisites for fertilization to occur. What is capacitation? It takes place in the sperm. Sperm capacitation is sperm acquires the ability to fertilize the egg through this process. Occurs while migrating through the female reproductive tract. Process in the sperm involves, that is, withdrawal of the cholesterol. As you can see, it is outside. All this, that is withdrawal. Then there is protein and glycoprotein substances on the acrosomal cap, that is there, that is also released, and this increases the removal of this cholesterol, that increases the whipping motility. So capacitated sperms, they penetrate the corona radiata, contact the zona pellucida, and they go the acrosomal reaction. So this is the acrosomal reaction. That means period of conditioning, or it is a prerequisite conditioning in the female reproductive tract, which take place in the males approximately for 7 hours and occurs in the uterine tube. Epithelial interactions between the sperm and the mucosal surfaces of the tube. What happens is a glycoprotein coat and seminal plasma proteins, they are removed from the plasma membrane that overlies the acrosomal region, and the cholesterol, as I showed you in the previous picture, that is removed. It becomes this process removal or the changes is what we call as the capacitation of the spermatozoa. This is a capacitated sperm which can pass through the corona cells and undergo the acrosomal reaction. So this is first is capacitation. Next step is going to be acrosomal reaction. Then next is your corona reaction, and then it is the zona reaction. There are different stages. What is the effect of capacitation? One is that it destabilizes the acrosomal sperm head. We said that the acrosomal was the glycogen, the Golgi apparatus that contains the lysosomal enzymes. That destabilizes this acrosomal head cap, which releases your lysosomal enzymes, which penetrate the zona pellucida. So it is the destabilization of the firm structure of the acrosomal cap of the sperm head that is destabilized, that releases the lysosomes which are inside the acrosomal cap, and they are released, and then they enter into the zona pellucida. So one function is this. The other is the chemical changes. They allow the increased mobility of the sperm tail. These are the two things which happen as a result of the capacitation. So capacitation is a prerequisite, and what happens is that the cholesterol is removed, the glycoprotein from the top is removed, and then you have the seminal plasma proteins. They are removed, and this leads to two changes. One is the destabilization of the sperm head, which helps in the release of the lysosomal enzymes from the acrosomal cap, and the other is the chemical changes that allow greater mobility in the sperm. Then there are three phases of oocyte penetration. That means before capacitation has occurred, now the sperm has to enter the oocyte. For that, three phases has to occur. In phase one, the spermatozoa must pass. Now, when the ovum, first we have to recollect when the ovum was released, what was all that was released? The corona cells which were released, which was the cumulus oophrus at the area which we call as the corona radiata cells. They come along with the ovum. We have the zona pellucida, which is there on the outside, and then we have the ovum. So this all has to be penetrated by the sperm to enter into the oocyte to form the pronucleus. So in phase one, the spermatozoa is going to pass through the corona radiata barrier. That is this outermost the cumulus oophrus. Then phase two is one or more spermatozoa penetrate the zona pellucida. Next layer is this red one. It has to pass through the zona pellucida, phase two. And the third one is spermatozoon penetrates the oocyte membrane while losing its own plasma membrane. So it's going to enter, it leaves the tail outside and it enters into the oocyte membrane, cell plasma membrane. So there are three phases, because what all is released out after ovulation consists of this corona radiata cells, zona pellucida, and then the ovum which is inside it. So the sperm has to pass through all these phases. So phase one is going to be corona radiata, second is going to be, as you can see in these pictures, it is passing through the zona pellucida, and third is meeting up through the plasma membrane. So it has to pass all these three barriers after capacitation to enter into the oocyte in order to fertilize it. Phase 1, penetration of the corona radiata. There are about 3 to 500 reach the site of fertilization. Only one is going to fertilize. Capacitated sperm passes freely through the corona cells. Dispersal of the follicular cells or the corona radiata cells, that is mainly the enzyme hyaluronidase from the acrosome of the sperm. This is the function, that is, it is dispersed by the enzyme hyaluronidase from the acrosome, which we destabilized in the the capacitation process. So that releases, I'll show you the pictures showing you the different processes. So that acrosomal cap has holes in it which releases this hyaluronidase enzyme, which is going to disperse these corona cells, and also enzymes which are coming from the tubal mucosal enzymes and movements of the tail. All these three things help the sperm to pass through this corona radiata or the corona radiata cells. So these three things, that is the enzymes, the mobility of the tail, and the motility of this tube, all these three things allow the sperm to pass, that is phase one, through this outer layer, that is the corona radiata cells. Okay. Then is stage 2. It has now to pass through the zona pellucida. So passage of a sperm through the zona pellucida is important phase in the initiation of fertilization. The permeability of the zona pellucida changes when the head of the sperm comes in contact with the oocyte surface. Lysosomal enzymes, this is going to change. These lysosomal enzymes which are there on the inside of the oocyte, that is going to uh, from the cortical granules on the inside, which are called the cortical granules lining plasma membrane. These, they are going to rupture and release certain enzymes which are going to allow the sperm penetrating the zona pellucida. This is what we call as the corona reaction. Once this corona reaction occurs, then these granules, what they are releasing, they are going to make changes in the zona pellucida. That zona pellucida change is going to make the zona pellucida impermeable. It is going to allow only one sperm to pass through the zona pellucida and make the other layer of the zona pellucida impermeable to the other sperm. So that is, it changes the permeability of the sperms and it prevents polyspermy, that is more than one sperm to pass through and fertilize the oocyte. That reaction is what we call as the zona reaction. So we have three I'm going to talk about in detail. One is acrosomal reaction, then there is the corona reaction, and then you have this cortical reaction, and then you have the zona reaction. Fusion of these oocytes, once they have passed through the zona, then through from the corona through the zona, you can see in this again, and then it's going to enter into the plasma membrane of the oocyte. The plasma or the cell membrane of the oocyte and sperm fuse and break down at the area of fusion. Head and tail, they stay out, but the sperm's plasma membrane remains behind, and then they enter. So we have, these are the three phases which are occurring. What is first thing is acrosomal reaction, by which it's going to pass through the corona and the zona pellucida. This occurs after binding to the zona pellucida induced by the zona proteins. This reaction culminates in the release of enzymes needed to penetrate the zona pellucida. Sperm penetrates the zona pellucida, and then a zona reaction occurs. That is three reactions: acrosomal reaction, corona reaction or the cortical reaction, and the zona reaction. Initially is acrosomal reaction, then there is a cortical reaction, and then there is the zona reaction. The zona reaction basically makes it impermeable, the zona pellucida to become impermeable. Now, what is the acrosomal reaction? The acrosomal reaction is the exocytosis of the acrosome, or the fusion of the acrosomal membrane with the sperm's plasma membrane. This process releases the acrosomal contents to the outside of the sperm and exposes the inner membrane to proteins onto the plasma membrane. So what happens exactly in acrosomal reaction, or how this is going to fertilize? I'll tell you, these are the steps which are occurring for fertilization. First is the sperm reaches the ovum, that is at the corona radiata level. Chemicals are released from the cells surrounding the ovum, triggering the acrosomal reaction. Then that is the beginning of the acrosomal reaction. Now what is going to occur? The acrosome of the sperm is going to swell, fuse with the sperm cell, and the surface membrane, and then these enzymes are released. Those are your lysosomal enzymes, or the digestive enzymes in the acrosomes are released. These enzymes, they are going to digest the follicle cells that is on the outside, and these follicle cells, so the sperm is going to pass through that layer. Once the acrosomal reaction ends, the zona pellucida is also penetrated by the help of these lysosomal enzymes. They make the pathway easy for the sperm to pass through. So basically, acrosomal reaction is going to help the sperm to pass through the corona radiata plus your zona pellucida. That is these two layers and they become soft and penetrable by the release of these lysosomal enzymes. So that is the pass. The sperm fuses with the ovum membrane. Now it's going to be the oval membrane. The sperm nucleus enters the ovum. Then starts the cortical reaction. These are the small granules which lie on the inner side of the ovum. These release certain substances. These released from the lysosomes in the ovum cause the zona pellucida to thicken, preventing the entry of another sperm. That is, polyspermy is prevented. That this release of the secretions is called the cortical reaction. Nuclei of the ovum and sperm fuse to produce, and so that change in the zona pellucida by this cortical reaction is what we call as the zona reaction, basically, which is the change in the structure of the zona pellucida. Nuclei of the ovum and then they fuse to produce a fertilized egg, that is the zygote. So these are the steps how the process of fertilization occurs. If you see in this diagram, first is this one, the sperm binds to the zona pellucida by the zona pellucida sperm binding protein. These are the follicular cells which are on the outside. This purple is your zona pellucida and this white is your perivitelline space. Then this acrosomal cap, it releases that is, we are occurring at the acrosome reaction. The contents are released by this exocytosis into the follicular cells and into the zona pellucida, right? So that means it allows the sperm to pass through these two layers. Then fusion of the membranes, hydrolytic enzymes help sperm to burrow through the zona pellucida, enabling binding fusion of the sperm, and then it is going to enter, passing through these two layers. Then it is going to help it to bind with the female nucleus. That is, sperm nucleus enters the egg. After passing through these two layers, haploid nuclei come together to form a diploid nucleus, forming the zygote. This is the basic diagrammatic procedure that is going to occur. You can see also in these two layers, the outer layer is the yellow one is the corona layer, and pink is the zona pellucida, and the inner single line is the cell membrane of the oocyte. How it is going to occur? Sperm activated by female reproductive tract that is going to come in contact with the zona layer. There, the zona, it's going to bind with it. It's going to be acrosomal reaction is going to occur. That is the release of these enzymes through this acrosomal cap, which allows the sperm to pass through this zona cumulus layer and the zona pellucida, and then it breaks into the zona layer. From the zona layer, it's going to penetrate. After passing through these two, it's going to release into the pass through this plasma membrane of the oocyte, and that enters, and there the male haploid and the female haploid gametes, they're going to meet together and become the diploid one. This picture also shows you the same acrosome containing hyaluronidase enzymes containing the nucleus come in contact with the vitelline layer, and this the zona pellucida. This is going to, one, contact and docking, then acrosomal reaction is occurring, release of these enzymes. This allows the passage through the zona pellucida, comes in contact with the perivitelline space, and then this sperm enters and leaving the tail and all the other structures outside, and then this comes in contact with the plasma wall, and then allows to enter inside and meet the, though become the male and the female pronucleus. So this is how the reaction occurs. That is, first you are producing capacitation outside, that is treating the sperm head to become changes to occur so that they can meet or release its enzymes. Then it is going to occur coming into contact with the ovum. That is, it needs acrosomal reactions, it needs cortical reactions, and then the zona reactions. These three are going to occur. Acrosomal reaction is why? Because we need the sperm to pass through this, the corona layer and the zona pellucida, and come in contact with the plasma membrane. Then this occurs, and then once they come in contact with the plasma membrane, the cortical granules, they are released, they pass out, they change the sperm has passed inwards, they change they further prevent the sperms to enter, so they change the texture of the zona pellucida, and they do not allow, that is cortical reaction, and that changes don't allow the further sperms to enter, and that reaction is what we call as, those changes is what we call as the zona reaction. So what is cortical reaction? Cortical reaction is a process initiated during fertilization by the release of cortical granules from the egg, which prevents polyspermy, fusion of multiple sperms with an egg. That is cortical reaction. If you see in this picture, this shows you acrosomal and cortical reactions together. So if you see this, that is the layer, the sperm, that is the acrosomal cap. When capacitation occurs, this head is comes in contact with the sperm outer membrane. Then this is released. There is exocytosis. It is released, and then it passes changes, makes changes in the corona layer and in the zona pellucida, and it allows the sperm to pass through, and it comes in contact with this ovum layer wall. There, these cortical granules, if you see, they are present. These release the secretions, lysosomal enzymes, these release, and they make changes in this zona pellucida, and this zona pellucida change is not going to allow any further sperm cells to pass inwards. This is cortical reaction, and then the sperm cell, is the oocyte meets the sperm. That is called they are going to swell up. That is called pronucleus, male and female. The tail and the rest of the structure is going to stay outside. It does not enter into the oocyte. So all this is again a repetition. This is the acrosome jelly. This is the jelly coat, that is zona pellucida. These are the receptors which stimulate or attract the sperm towards the ovum. Then this acrosome, it is going to release these enzymes. These enzymes, they're going to produce the changes in the zona cumulus layer and in the zona pellucida, pass through inside, coming in contact with these cortical granules. If you see it in the picture, then this is going to release the head inside, that is, which has the nucleus, which has the DNA material, that is going to come inside to the oocyte, and the tail and the rest of the structure stays out. And you can see these receptors, they're going to release these cortical granules, the material, and they pass into the zona pellucida, and they prevent further fertilization. That is, they do not allow further sperms to pass through. So all these three reactions are going to occur simultaneously when the sperm, it passes inwards. So basically, after capacitation, we have done acrosomal reaction, cortical reaction, and then zona reaction. Zona reaction is nothing but it refers to a modification of the zona pellucida in response to fertilization, which alters the solubility and binding properties of the zona pellucida and leads to a slow block to polyspermy. So that just one sperm enters. Many have reached the level of the oocyte, but one is going to just fertilize, and that blocking up is done by this zona reaction or the cortical enzymes, which make the changes in the zona pellucida, and that is, it prevents polyspermy, and that reaction is called zona reaction. So then this is what events occur in fertilization. First, it was the ovary. The ovum is released, the fallopian tube, it sucks in, and then these are the changes, that is the entry of the sperm into the ovum. Sperm and the oval nucleus swell, forming the pronucleus, as I said, both. Then DNA replicates because both was haploid, and the spindle forms between them. The fertilization is accomplished, zygote is formed, spindle formation, and then that is the meiotic. If it is fertilized, the meiotic, second meiotic division is going to continue, and then there is spindle formation, and then the diploid cells are formed, which is the zygote is formed by the meeting. These changes are going to occur. That means when sperm and the oocyte meet, they are going to form the pronucleus, swell, and then they are going to have enter into the second meiotic division, which was standard until and unless fertilization occurs. Then the fertilization is completed and it is a diploid zygote is formed. So thus, if you see the sequence of events of the reaction is acrosome reaction, cortical reaction, and zona reaction. Zona pellucida attracts sperms. Sperms bind to the membrane. Acrosome, release of enzymes. Penetration of sperm into the zona pellucida. Fusion of sperm membrane with the oocyte membrane. Cortical reaction. Release of the cortical granules, which produce this changes in the zona pellucida. That is, and that permeability change is what we call as the zona reaction. So these three occur after capacitation. Capacitation was changes in the sperm itself. That is, the cholesterol is removed, the head proteins are removed, and the glycoprotein changes occur, and it makes it destabilized. That means it can release the enzymes which are present in the acrosomal capsule, and which is going to release and enter and change the texture of the structure of the zona pellucida and the corona, so as the sperm can pass through these two layers. And that is, and it also increases the mobility of the tail. So these are the results of the capacitation, and then this process is going to occur and it forms the fertilization. If you see again, this picture is also going to tell you A is attachment, then it is going to bind, then the acrosomal reaction is going to occur, it penetrates. Green is zona pellucida, and outside you have this. Then there is fusion. These are the cortical reaction. If you see that they are released, they make the changes. The started is the zona reaction, this zona reaction, and then they are going to prevent the rest of the sperms from entering. So you can see they are stopped and polyspermy is blocked. And then this was the first polar body, and then this the two, they are going to meet, and then there is going to the second meiotic division is going to complete, and then the zygote is formed. So this is a complex collection of the three phases which are going to occur, and all the, so this picture shows you all the steps which are taking place in fertilization. As soon as the spermatozoa has entered the oocyte, the egg responds in three ways. Cortical and zonal reactions occur. The oocyte membrane becomes impenetrable to the others as spermatozoa and the zona pellucida outer, so it prevents polyspermy. Then there is the resumption of the second meiotic division. As I told you, oocyte finishes its second meiotic division, which results in a second polar body and the definitive oocyte, that is 20. This nucleus is also known as the female pronucleus. This second change occurs. Then there is another change is metabolic activation of the egg. Initial cellular and molecular events are associated what is going to be the further division changes which are going to occur in the embryogenesis. Those take place. Then there is the formation of the female male pronucleus within the cytoplasm of the oocyte. The nucleus of the sperm enlarges to form the male pronucleus. The tail is going to degenerate. It does not come inside. Male and female pronucleus are then going to come close together, contact and lose their nuclear envelopes. Both are haploid at this stage. This two meeting of the both haploid male and female structure which is formed is called a pronucleus, is called an ootid. Each pronucleus must replicate then its DNA. Then it has to replicate. Then the chromosomes organize on the spindle for a normal mitotic division, 23 maternal, 23 paternal. Chromosome ootid becomes a zygote. So that means the division starts and then they split up longitudinally at the centromere. Sister chromatids moves to the opposite poles, diploid number of chromosomes and DNA is formed. Then there is again all that the furrow on the surface of the dividing the cytoplasm into two parts. So haploid becomes diploid, it enters into the second meiotic division. So if you see in this picture, this is entered, this is the spindle second maturation division, then it increases in size, the pronucleus that produces the secondary oocyte, and this pronucleus and the polar bodies. Then there's the centrosome, again the division starts, and this is the then mitotic division starts. This completes the meiotic division, and then the mitotic division starts, and it starts to divide the cell from 2 to 4 and all words. That is the cleavage process starts. If you see in this picture again, this is going to show you the different stages. The sperm has entered oocyte with simple set of chromosomes, that is N. They are going to merge together, producing the pronucleus. Then this is going to combine together, and then the meiotic division, it's going to multiply before that into the doubling of the DNA, so it becomes 2N, and then they are going to start its cleavage division. So the second meiotic division is going to complete, DNA is going to double itself, and then the meiotic division, that is the cell division or the cleavage process or the morula formation, that starts. So fertilization is a complex sequence of events. Contact between a sperm and an oocyte ends with mixing up of the maternal and the paternal chromosomes at metaphase of the first mitotic division of the zygote. Occurs in the ampullary region of the uterine tube. What are the results of fertilization? What happens when once fertilization has occurred? It stimulates the penetrated oocyte to complete the second meiotic division, as we've said. Restores the normal diploid number of chromosomes is restored. That these are the results of fertilization. Then mixing up of genetic material leads to variation of human species through mingling up of maternal and paternal chromosomes. Then the chromosomal sex, once the fertilization has occurred, chromosomal sex of the embryo has been determined and causes metabolic activation of ootid and cleavage, that is the mitotic division starts, that is cleavage starts. So all these steps occur once fertilization has occurred. That is the two pronuclei meet together and then they go through all these changes. That means this is going to automatically result. Completes the second meiotic division, restores, that is by that it is diploid number of chromosomes, doubling of the DNA material, diploid number is produced, variation, mitotic cleavage starts, because then the cells have to go through the mitotic division. So once meiotic division is complete, second is finished, the DNA doubles itself, and then again the mitotic division starts, and then the cells from divides into one, divides into two, and onwards. That is the cleavage starts. And what is the result? Genetic mixing up is there, chromosomal sex is determined, metabolic activation of the fluid, that is the mitotic division starts. Zygote is genetically unique. Half of its chromosomes is from the mother and half is from the father, a new combination. So it is said it is a biparental inheritance and variation of human species. That is, it is a mixture up of chromosomes. That is why it is said The inheritance is from both the parents. Crossing of the chromosomes, relocating segments, maternal and paternal, shuffling of the genes is there. And so there is a genetic recombination that is there. Inheritance is from both the parents, and then there is shuffling or crossing over of the genes, that is genetic recombination occurs. The embryo's chromosomal sex is determined at that time, fertilization by the kind of sperm X or Y that fertilizes the oocyte. So fertilization, because it has X and Y. So fertilization by X bearing sperm produces a zygote, XX, a female, and if it is by Y, it is a male. Then meiosis allows, that is half of the genetic material, independent assortment of maternal and paternal chromosomes among the germ cells. That is the change that is there. So that is how fertilization is going to occur, and what all changes occur when fertilization occurs, and what are the prerequisites which should occur, and what are the changes which are going to occur through fertilization. What is early pregnancy factor? How early can this pregnancy be detected? It is an immunosuppressant protein. It is secreted by the trophoblastic cells, that is outside, and they appear in the maternal serum within 24 to 48 hours after fertilization. So it is said that this forms the basis for the pregnancy test during first 10 days of development, until and unless it can be seen on the ultrasound. Pre-selection of the sex, how it can be done. X and Y sperms are formed in equal numbers because the division is there, so it is expected that there should be 100 girls and 100 boys, but various techniques are used to make up their selected sex, whether they want a baby girl or a baby boy. So they separate the X and Y sperms. That is called gender selection. This process is called gender selection, when you separate out X and Y and you select and make a combination of XX or make a combination of XY. That is gender selection. By your own choice, you produce a baby girl or a baby boy. This is by the different features of the X and Y. They are the differential swimming abilities of the X and Y, speed of migration of the sperms, difference in appearance, DNA difference, all these things. They recognize that this is X and this is Y, and then they mix up and then they produce the selected sex. The use of selected sperm sample is in artificial insemination may produce the desired sex. That means you recognize with that and then you cross over the required genetic material. What is infertility? Infertility is problem for 15 to 30% of the couples. Male may be a result of insufficient number of the sperms, or they are morphologically not normal in structure, or the motility is poor. All that leads to male infertility. Normal ejaculate of the sperm is about 2 to 6 ml, 100 million per milliliter, or 20 to 50 in total ejaculate as are usually fertile. Infertility in women is because of the uterine tube problem, hostile cervical mucus, immunity to spermatozoa, or absence of ovulation and other features. All this leads to the infertility in females. So there are certain methods that we can use for assisted production. That is all what is the test tube babies and all that that is used. That is what is called as assisted reproductive technology, ART. Offspring from these conceptions show you increase in prematurity because they are born earlier, then low birth weight, maybe because of this artificial way, the birth weight. These are the problems that are seen because of this assisted reproductive technology. Premature babies, smaller in size, and motility is high because they do not reach till the end because they're all artificially maneuvered. Outcomes are caused by increased rates of multiple births. As I told you before, when they're twins and triplets common in RT pregnancies, because when you artificially stimulate the ovulation process or the production of the follicles, more than one is start to produce at one time, so you can have multiple births. Among the singleton births, low birth weight and mild form infants can be there. So it's something which is artificially done. Many problems are there. Assisted reproductive technologies. What is that? In vitro fertilization and embryo transfer. In vitro fertilization is of the oocytes and transfer of cleaving zygotes into the oocyte. Opportunity for many would bear child. Embryo is the fertilization is done in outside, then the cleaving. Then once the fertilization is done, the division cleavage starts. That is that at that stage the zygote is born into the or put into the female genital tract, and this can standard procedure by laboratories throughout the way. That is fertilize it outside on the Petri dishes. Once the cleavage starts, fertilization goes on, and the cleavage process starts, then it is put in the mother, and then this process of pregnancy goes on. That is in vitro fertilization. That is outside. The steps in vitro fertilization is ovarian follicles are stimulated by the drug what we called as citrate, clomiphene citrate. Many oocytes are aspirated by laparoscopy. Oocyte placed in dish, special cultured medium, and capacitated sperms are put there. Fertilization occurs, cleavage of this process starts, then resulting into 4 to 8 cell stage that is put back into the uterus, and the patient has to lie for several hours. This chances of multiple pregnancies, spontaneous abortions, of transferred embryos is also higher than normal. So that is the process of in vitro fertilization. That is, fertilization is done outside to make sure that the fertilization process is there, and then they're put back in the mother's uterus and then allowed the process to go on. That is what is called the in vitro fertilization. And see, in this process, ovaries are sucked artificially by laparoscope. The fertilization is done on Petri dishes, and then it is put back in the uterus. Success rate, maternal age 30% and more will conceive with one attempt, less, and if younger than 35, and if rate drops to 20 will less, if the over age 40%. More chances of congenital malformations are there. Multiple births are a disadvantage, have a high rate of morbidity and mortality. Male infertility, oligospermia, that is no live sperm. Azoospermia is few. Oligospermia is few live sperms are there. Azoospermia is no live sperms are there. That is the reason of infertility. Intracytoplasmic injection, overcoming using intracytoplasmic sperm injections which are there. A single sperm is injected into the cytoplasm to the egg to cause fertilization offers. So sperms are taken out. These are injected into the egg, causes fertilization, and the couples to or a donor sperm can be used. If the male sperm is not present, this is then injected into the mother, into the cytoplasm of the egg, causes fertilization outside. The fertilization process is done outside the body, and then made sure once the fertilization has occurred and the cleavage process starts, then they are injected into the female, or they're placed into the female genital tract. Then there are cytopreservation of embryos. Different other processes are there. Embryos resulting from in vitro fertilization can be preserved. Fertilized embryos can be preserved for long periods by freezing them with a cytoprotectant. Transfer of 4 to 8 cell embryos and blastocysts to the uterus is now common. That means after frozen fertilized embryos, they can be thawed and placed back into the mother. Intracytoplasmic sperm injection can be injected directly into the cytoplasm of the oocyte, used for treatment of couples. In vitro fertilization has failed and too few sperms, so directly. Assisted vitro fertilization, that is called GIFT. The term is gamete intrafallopian transfer, that is done at the level of the gametes. A technique fertilization to occur in uterine tube is called gamete intrafallopian transfer. So abbreviation is GIFT. It's not the other gift that you give. It involves superovulation, releasing of many of the ovas are removed, sperm is collected, laparoscopic placement of several oocytes and sperms into the uterine tube. So you remove ova and sperm, you place them in the fallopian tube, and you are producing, this is at the level of the gametes, and you produce this fertilization in the ampulla. It's the usual location. This is a gamete intrafallopian transfer. So you transfer the two gametes into the intrafallopian area where you make the fertilization to occur, and then the process to go on. So that is called as GIFT. Then there's something what is called as a surrogate mother, a mother who doesn't have a uterus. So she needs, or the uterus ball is not normal, she needs somebody to carry the baby for 9 months. So fertilization occurs, and then this fertilization, the fertilized baby is transferred into some other mother's uterus, and she carries the baby till 9 months, and when the baby is born, it is handed over to the original mother. That is surrogate mother. That woman who is carrying somebody's baby is called a surrogate mother. Produce mature oocytes but are unable to become pregnant because his uterus has been excised. So in that case, embryos are transferred to another uterus for development and delivery. Then the gamete intrafallopian transfer, similar to intravitreal, this produces fertilization naturally. Doctors places the egg and the sperm directly into your fallopian tube. Gamete uses multiple eggs collected, placed into a thin tube. These gametes are then injected into the fallopian tube using a surgical procedure, will give general anesthesia. So they implant that. Then there is something what we call a ZIFT, that is the zygote intrafallopian. There we were transplanting, the working with the gamete. In this, we are working with the zygote. And infertility treatment used when the blockage in the fallopian tube prevents the normal binding of the sperm into the egg. Eggs are removed from the woman ovary and in vitro they are fertilized. The resulting zygote is placed into the fallopian tube by the use of the laparoscopy. So in this, when you have a problem in the fallopian tube, you cannot start the procedure by these gametes. So what you do is you fertilize the move from the woman's ovary and they are vitro fertilized, that is they're fertilized outside, and then they are placed in the fallopian tube, and from where on they by the laparoscopy, and then from there on they are going to start the procedure. So in that you were implanting the gametes, we were working with the gametes, that was GIFT, and ZIFT is the zygote intrafallopian transfer, that is you are working with the zygote. You have are done in the way you do it in the test tube babies there, intravitreal fertilization. You fertilize and then you place this in the fallopian tube, because then you are sure that this fertilization has occurred, and then you can place it in the area other than the ampulla of the fallopian tube. That is ZIFT. Intrafallopian combines intravitro and the GIFT. That means both the processes are collection. Stimulated and collected in one method, eggs are mixed with sperms in the lab. Fertilized eggs are then laparoscopically returned to the fallopian tube. So in this, the zygotes are put back. In that, it was the gametes were carried by the uterus developing into a fetus. Then there's certain contraceptive methods, which are bariatric. They're condoms, male, female, diaphragm, cervical caps, sponges, contraceptive pills. Basically, the function of the pills is that it prolongs the time period for, or it interferes with the changes of the hormones which are going normally into the uterus. Depo-provera implanted somewhere, which gradually occurs, prevents ovulation. Then when the certain causes abortion administered within 8 weeks of menses. So there are different ways which are produced. So male pill contains synthetic androgen, prevents luteinizing and FSH secretions, either stops the sperm production and reduces the to a level of infertility. So different ways of action. Intrauterine device, that is produced, the mechanism preventing pregnancy is not clear. Inhibition of pre-implantation of stages, that means it prevents the implantation of the ovum to occur. So it interferes with the changes which are occurring in the uterus, and it does not allow the implantation to occur. Vasectomy and tubal ligation both produce a reversible, that is either you tie up the fallopian tube or you tie up the vas deferens, and then you can even open them up, and that really leads to the normal process again. So this is the implantation. We'll talk about it next time. So that was about the motility, how the change cells are fertilized, gametes are fertilized, what is infertility, what is the different procedures of the fertilization which occur. Now, once the fertilization has occurred, what is going to occur? The mitotic division has started the cleavage. So gametes were there, entered male and female pronucleus. These male-female pronucleus, they were haploid. They completed the meiotic division, and this meiotic division then changes on to the doubling the DNA material, and this double of the DNA material is going to start the cleavage material. That means mitotic division is going to start, and this is going to lead, what is this showing this picture, that is the two-cell stage, that is the mitotic division is going to start. So, and then once this starts, then there is going to lead on to this from the fallopian tube. It is going to move on into the uterus, and where the implantation is going to occur. What can lead to the problems, what implantation is there, what changes are going to occur as the morula or the ball of cells is transferring forward. So in the next class, inshallah, we'll complete this implantation. Okay then. Thank you.